practical, you were supposed to be carrying out pore plating and spread plating. So just to refresh your memory, for pore plating, you are given a new petri dish plate. You add your sample here, okay, and then you pour your molten agar in. Okay, so once you pour your molten agar in, usually we only pour about 3/4 full, and then we will swirl it, which means we'll mix it, turn it 3 times clockwise, then 3 times anti-clockwise, 3 times up and down, 3 times to the sides. Because we have to make sure that your sample is well mixed with the agar. Then we will allow the agar to set together with your sample inside. Then when it's hardened, we will incubate it in the incubator inverted. Okay, we invert the plate when we incubate in order to prevent condensation from forming onto the cover here. Because if for example, if you were to incubate it like this, okay, and there is condensation here, water, this will increase the chances of contamination. That is why we incubate plates inverted, okay, to prevent or to reduce chances of contamination. So for pore plating, you are given an empty petri dish plate. You have to put your sample in, then add about 3/4 of agar, molten agar, and then swirl it 3 times clockwise, 3 times anti-clockwise, 3 times up and down, 3 times to the side to ensure homogeneity. So ensure well mixing. Okay, ensure well mixing because this will determine the accuracy of your count. Okay. It's different approach. So your plates are already prepared. Okay, your plates are already prepared, already solidified. You prepare a zero dilution of your sample, then place your sample onto the agar, and then you use a spreader, a hockey stick spreader. Okay, usually this is glass spreader. You sterilize it with alcohol and flame it before you spread the inoculum around the plate. So you are spreading the inoculum around the plate. Okay. So if you notice, you ensure good distribution here. Okay. So this is the same as when you're doing pore plating. You ensure homogeneity or well mixing or good distribution. Okay. Because you are counting colony forming units. The assumption here is a single bacterial cell will give rise to a colony. So a single bacteria will give rise to a colony. Okay, that means you have to make sure your cells are well distributed, well spread out, okay, homogeneous across the plate. If you have cells clumping like this and they form a colony, then you are underestimating your bacterial count. Okay, that is why this is critical. Good distribution and homogeneity of your sample. Okay, on the assumption that a single cell will give rise to a single colony. So because in culturable bacteriology we are measuring colony forming units. Okay, CFU. Okay, so that's why we have to ensure a good distribution here. Since your spread plate, the bacteria is spread onto the plate, your colonies will form on the surface. But for pore plating, since you're mixing the sample with the agar, your colony will sometimes grow inside, most of the time, grow inside the agar. So there is a essential difference here. So which is better? It depends on what you need. So if you are working with spread plate, then it'll be easier for you to further isolate your bacteria. Here it'll be harder because you have to break through the agar. Okay. Another difference here is in spread plate, you use a physical force, a stress. Physical stress where you use a glass rod onto your sample. Whereas here in pore plating, there is thermal stress because the molten agar is around 40 to 50 degrees Celsius and may kill some bacteria. Okay, so there are differences between spread plating and pore plating. The inoculum used in pore plating is usually 1 ml, whereas for spread plating is usually 0.1 ml. If you put too much inoculum into your spread onto a spread plating technique, you will find that your bacteria will tend to settle at the side and make and grow as a large boundary. Okay, because the bacteria the sample will if your sample is too much, your plate will be too wet. So even when you try to spread it, it will not work that well, and most of your samples will go to the sides. Okay.